Red-green colour blindness and the salt pans of the Dead Sea. No obvious connection between them. And yet the light-sensitive pigments of the human eye share striking similarities with a salt-loving bacterium. It's a tale of variations on the theme of cells. Red-green colour blindness affects 8% of men and 0.5% of women. In recent years, we've come closer to tracing the basis for this condition. Enter the first set of characters in our tale, the light-sensitive cells in the vertebrate eye. The larger ones, called rods, function in dim light and are responsible for black and white vision. The smaller tapered ones, called cones, work in bright light and are responsible for colour vision. Isolating these cells calls for some nimble surgery. Cutting through the tough outer wall of the eye, we see the iris and the clear lens. Behind the jelly-like vitreous humour is the retina, a thin tissue of light-sensitive cells. In this ox eye, portions of the retina are highly reflective. The tissue is attached to the back of the eye, passing out through the optic nerve. Studying vision often entails working in the dark, or at least in dim red light, to which the retina is relatively insensitive. Switch on an ordinary light, and the effect on this frog retina is obvious. We can even follow this response in individual rod and cone cells. Jim Bowmaker at the Institute of Ophthalmology in London.
When you come to look at individual cone cells, they're very small, of course. They're only a micron or two in diameter. The pigment within them is photosensitive, so by their very nature they're difficult to study. The moment you shine a light beam on them, you're liable to destroy the visual pigment you're trying to measure. However, if you use very dim light beams, you can pass these through the cell and then measure the absorbance of the visual pigment.
The trick is to use a microspectrophotometer, which focuses two narrow beams of light. One passes through a cone cell. The other, called the reference beam, focuses on a clear area of the sample. The instrument scans through the spectrum and compares the intensity of the reference and sample beams at each wavelength of the incident light. This cone responds to light from all parts of the spectrum, but is most sensitive to long wavelength reddish light. The beam is lined up on another cone. This time, it absorbs most in the middle wavelength or green part of the spectrum. Most of the cones in the retina are either red or green sensitive in roughly equal numbers. But the cones that are most sensitive to the short wavelength or blue region of the spectrum are much rarer. Only about 1 in 10 of the cones in the human retina are of this type. So human colour vision is trichromatic. It's based on 3 types of colour receptors. It's the integration of information from these 3 channels that allows us to see a full colour spectrum. So what happens in the case of red-green colour blindness?
Right, Colin, in this colour vision test we have 15 little pots, each one with a coloured disc on top. And what you have to do is put them in sequence, starting with this fixed pot at the end, and match them as close as you can to each other. It doesn't matter how long you take, and you can switch pots as often as you like. So I'll leave you to it. Red-green colour deficiencies in man fall into 2 groups. There are some individuals who are completely red-green colourblind, and that is that they've lost one of the types of photoreceptor in the red-green part of the spectrum. They may either be absent of the red pigment or of the green pigment. In that way, they cannot distinguish that part of the spectrum. In some cases of colour blindness, normally called anomalous colour vision, people are trichromatic, that is, they have three colour channels. One of the ones in the red and green part of the spectrum is spectrally displaced. This means that their colour vision is going to be rather subtly different from normal observers. The underlying molecular basis of this appears to be due to changes in the protein structures of the visual pigments.
It turns out that the light-sensitive pigment in rods and cones is a derivative of vitamin A called retinal. In this particular conformation, it's known as 11-cis retinal. In rod cells, the retinal is associated with a protein opsin to form a complex known as rhodopsin. A similar complex is found in cones. The three-dimensional structure of these visual pigments isn't known precisely, but intriguing clues have emerged from a quite unexpected source. The Dead Sea may seem an unlikely haven for most forms of life. Salt concentrations may be 7 times that of normal seawater, but certain organisms thrive here. The salt lovers or halophiles. These tiny bacteria actually require high concentrations of sodium chloride to survive. They're known as halobacteria. In the absence of oxygen, halobacteria switch on a primitive form of photosynthesis. Large patches of a special protein, bacteriorhodopsin, accumulate in their membranes. What's the role of this protein in the cell? Richard Henderson, from the Laboratory of Molecular Biology in Cambridge.
Bacteriorhodopsin is a light-driven proton pump, so that its function is to absorb light and then use that light energy to pump protons from the inside to the outside of the cell. The cell then has a built-in proton gradient across all parts of its membrane, and that proton gradient is an energy source that can be used for all sorts of other things, like making ATP, pumping in or out ions or other small molecules like amino acids, or powering the rotary flagellar motor that makes the bacteria swim around.
Unlike most membrane proteins, bacteriorhodopsin is relatively easy to isolate. Place the bacteria in distilled water and their cell walls disintegrate. The released membranes can then be separated out by centrifugation, forming a series of coloured bands. The orange-red layer contains the pigment bacteriorubrin, a sort of bacterial sunscreen that protects the cells from the harmful effects of sunlight. The other band contains a set of purple membranes. It's these that contain the protein bacteriorhodopsin. Now, isolating a protein is one thing. Determining its three-dimensional structure is another. Membrane proteins have proved notoriously difficult in this respect.
There were very few of these proteins available pure and in a form ready to study, and one of them was the bacteriorhodopsin molecule from halobacteria. You could grow the bacteria in your lab very easily. The protein was so plentiful in the bacteria that it filled about half of the membrane, and the concentration was so high in the membrane that it also formed a two-dimensional crystal. But it turned out that even with the two-dimensional crystals of the size they come in vivo in the bacteria, were quite suitable for the sorts of electron microscopy methods which we've used for analyzing their structure. We put the specimen in the electron microscope and illuminate it with a parallel beam of electrons. We then take perhaps 100 different images of the same membranes, illuminated from different angles by the electrons. We do that by tilting the specimen in the electron microscope up to perhaps 60 or 70 degree tilts. We then get a collection of images and recombine those to regenerate a three-dimensional representation of the scattering matter in the crystal, which contains the molecules we're interested in. And by doing that, we can then get a map of all of the atoms in the structure that contribute to each image.
But how did they convert this contour map into a model of the protein? This involved the use of computers in a sophisticated task of molecular model building. The results were striking. A cluster of structures commonly found in proteins, alpha helices.
In the map that we have, which shows the density that makes up the structure, we can look into it and interpret it not only in terms of the alpha helices, which are gross features in terms of some people call secondary structure of the protein, but also in terms of the smaller groups of atoms that make up the amino acid residues. That way we get a model for the whole structure, which then, because we know the amino acid sequence, tells us a bit about the chemistry of the environment of the retinal pigment and the inside of the protein versus the outside where it's in contact with the lipid.
In the current model for bacteriorhodopsin, the protein lies embedded in the lipid bilayer, with the alpha helices extending from one side of the membrane to the other. The start, or amino terminus, of the protein chain lies outside the bacterial cell, while the tail, or carboxy terminus, lies in the cytosol. There are, in fact, 7 helices running sequentially along the chain, linked by short loops. Together they form a channel in which the pigment retinal is located. We can pinpoint some of the amino acid residues that come into close contact with the retinal. For instance, there's a specific lysine that's chemically linked to the pigment. Then there are aromatic amino acids, such as tyrosines and tryptophans. There are also several negatively charged aspartates. We can see these interactions more clearly in a computer graphics model. The aromatic residues form a water-repelling or hydrophobic pocket that holds the retinal parallel to the membrane, the best orientation for it to absorb the light. The charged aspartates play a role in the proton pumping function of the complex. So what happens during a single photochemical cycle? Here's one possible model. When a photon of light is absorbed, the retinal molecule changes conformation. A proton gets transferred from the lysine to one of the aspartates and then to the outside of the cell. This proton is replaced by another coming from the cytosol, possibly via a second aspartate deep within the membrane. The cycle can then be repeated. The net result is that one photon of light causes one proton to be pumped from the inside to the outside of the cell. In fact, there's a build-up of protons across the membrane, so that the bacterium is charged with a proton gradient that acts as a source of energy. So the molecular structure of bacteriorhodopsin is giving powerful insights into how the protein acts as a proton pump. But the bacterial story doesn't end with proton pumping.
So bacteriorhodopsin, under the conditions where it's needed, might occupy half of the cell membrane. But at a much lower level, 50 or 100 times lower, there are other pigments that have other functions. One of them is halorhodopsin, called because instead of pumping protons out of the cell, giving you an energy source for the cell, it pumps chloride ions into the cell, and the cell needs to maintain a good balance of the ions, sodium and chloride, potassium and so on, for its particular halophilic environment. So there is another light-driven chloride ion pump that is responsible for maintaining the chloride ion balance of the cell. Then at even lower concentration, there is a tiny amount of another retinal protein called sensory rhodopsin, and its function is to absorb light and then somehow, by a mechanism that isn't known yet, to allow the bacterium to sense where the light is. So it's a phototactic pigment.
Bacteriorhodopsin is the only one of the 3 pigments that we have a detailed 3-dimensional structure for. Its 248 amino acids have been fitted into a 7 helical model like this. The 20 or so residues important in retinal binding form a band in the lower half of the molecule. Both halorodopsin, with 251 amino acids, and sensory rhodopsin can be fitted into the same basic model by insertions or deletions in the loops between the helices. It turns out that around 30 amino acid residues are identical across all 3 proteins. This corresponds to a 15% level of identity or homology. Over half of these are in the region of the retinal binding pocket. So the region of the protein that contains the retinal binding site is highly conserved. But what does this family of bacterial proteins tell us about the visual proteins in humans? The rhodopsin in rod cells, and the blue, green and red receptors of the cone cells. It was in comparing mammalian rhodopsin with bacteriorhodopsin that some structural relationship emerged. Knowing the 3D model of the bacterial protein and the amino acid sequence of rhodopsin, it was possible to predict the secondary structure of the visual pigment. The result was a surprise. It fitted into a model of 7 helices, just like bacteriorhodopsin.
Both of them have 7 helical sections of the polypeptide, 7 transmembrane helices. Both of them have their amino end on the outside of the cell. They have their carboxy end on the inside of the cell. Both of them have the pigment retinal covalently connected to a lysine halfway along the 7th helix. So there are these then circumstantial structural evidences that they're actually more similar than their different functions would suggest.
We can extend this sort of analysis to the other members of the visual opsin family. It turns out that about 40% of the amino acids in each of the cone receptors is identical to rhodopsin. So it's highly likely that these fold into a 7 helical structure as well. Using this model, what differences do we find between the green and the red receptors? It turns out that they differ in only 15 of their 350 amino acids. Of these, only 3 are close enough to have any effect on the light-absorbing properties of the retinal. In one case, an alanine in the green receptor is a threonine in the red receptor. In the other 2, a phenylalanine in the green receptor is a tyrosine in the red. These subtle differences may be enough to alter the environment of the retinal and shift its absorbance peak in the spectrum. Subtle differences, but a big enough shift for the human eye to distinguish green from red. These predictions about the human visual pigments have come from models based ultimately on a protein from bacterial cells. How valid is this comparison likely to be? Although bacteriorhodopsin itself isn't involved in vision, remember that a closely related protein within the bacterial family is. This is the phototactic pigment sensory rhodopsin.
Its function then is really identical as far as the bacterium is concerned to visual rhodopsin in mammals. It's the bacterial eye, if you like. It allows it to swim around, see where the light is and swim into it, but in a much less intelligent way. So there is a bacteriorhodopsin homologue, the sensory rhodopsin, which is completely related at the level of amino acid sequence to bacteriorhodopsin and related at the functional level to mammalian rhodopsin. So although there isn't an unbroken chain of logic linking the 2 families together, there is the circumstantial evidence on the structure there is the amino acid sequence connection between bacterial rhodopsin and sensory rhodopsin, and the functional relation between sensory rhodopsin in bacteria and visual rhodopsin in mammals. So people are now much happier with the idea that although they are two separate families, probably they are sufficiently closely related that when we know the structure of the rhodopsin family, we'll find that it's very much the same as bacteriorhodopsin.
On the face of it, you couldn't think of two cells less alike than a salt-loving bacterium and a cone cell in the human retina. Yet, as our tale of two cells has shown, there is a very interesting connection between them. Both exploit membrane proteins that contain retinal, and so put sunlight to cellular use. Now, of course, this is a very peculiar connection, but it makes a good story, and that's why we chose it. But all cells, however different, do have certain features in common. All living cells can be regarded as evolutionary variations on a theme, or set of themes. Now, if you're studying cells, this is very useful. Understand the underlying themes, and then you can much more easily investigate the vast range of variations that evolution plays upon them. Now, just for a minute, let's go back to where we began the series, with our Martian trying to understand how cities work. It wouldn't really matter which city the Martian understood. Having worked that one out, he or she could be put in any other city and they'd soon feel at home. Might not immediately understand the function of a particular building, but would recognize it as a building and would have some idea of how to investigate its function. And that's the key, relate structure to function. The advice we gave to the Martian at the outset, and advice that we have adopted fruitfully in investigating living cells. Of course, we don't know everything about cells. There's a lot more exploration to be done, and I'm sure there'll be a few surprises in store. But there's one thing on which I'd place a large bet. The more and more closely we look at living cells, the more we'll realize how precise, how elegant the fit is between structure and function. So, if you're really interested in the subtleties of the connection between structure and function, forget about cities, leave them to architects and Martians. In fact, leave the analogy behind. Look at living cells.